Welcome to Ultimate Sleep Doctor. Tonight, we'll gently explore a hidden world, a realm of quiet wonders. Beneath our feet, within shadowed forests and damp earth, exists a kingdom of fungi, a vast, intricate network unlike any other. These organisms, often unseen, are architects of soil, recyclers of life, and possess a silent beauty. They've existed for hundreds of millions of years, observing the slow dance of time. It's a reminder that there's more wisdom and resilience in the natural world than we often realize. If you find yourself drawn to the quiet mysteries of the earth, we invite you to subscribe and share this space with others. Let your thoughts drift. Just breathe. Let's begin. Deep beneath the mossy carpets of the ancient forest, a secret world hums with activity. It's not a world of creatures you can easily see. No bustling deer or chattering squirrels occupy this space. Instead, a vast, intricate network spreads out beneath our feet, a silent, sprawling intelligence composed of threads finer than a human hair. This is the mycelial network, and it's quite simply the largest living network on Earth. It's a hidden internet, a communication system, and a remarkably efficient delivery service all rolled into one. The mycelium itself is the vegetative part of a fungus. Think of it as the roots of a mushroom, but vastly different in form and function. It doesn't anchor a mushroom to a single tree or rock. Instead, it's a branching, interwoven mass of hyphae. Hyphae are individual filaments, like tiny threads, and they're the workhorses of the network. These hyphae grow outwards, exploring the soil, seeking out nutrients, sugars, phosphates, nitrogen that the fungus needs to thrive. But their reach goes far beyond simple feeding. They form a complex web, connecting with the roots of countless trees, plants, and even other fungi. The beauty of the mycelial network lies in its ability to facilitate symbiotic relationships. Many trees, particularly those in old-growth forests, engage in a mutually beneficial exchange with the fungi. The trees, through photosynthesis, create sugars as a byproduct of their life. They pass a significant portion of these sugars up through their roots and into the fungal network. The fungi, in turn, provide the trees with access to water and nutrients that might otherwise be locked up in the soil. And crucially, they extend the tree's root systems, vastly increasing their absorptive area. It's a sophisticated sharing system, a trade agreement orchestrated by a silent partner beneath the surface. But the network's capabilities extend far beyond simply transporting nutrients. Scientists have discovered that the mycelium uses electrical signals to communicate with other parts of the network and even with other organisms. These electrical signals aren't like the ones our brains use. They're much slower, operating at a rate of perhaps a few milliseconds per signal. However, they travel incredibly long distances through the network, far exceeding the reach of chemical signals. Researchers have observed electrical pulses traveling hundreds of feet along fungal hyphae, demonstrating a form of rapid communication. The exact nature of these electrical signals is still being investigated, but it's believed that they are linked to resource allocation. When one part of the network senses a need for water, for instance, it can send an electrical signal to another part that is rich in water, triggering a flow of water along the network. Similarly, if a tree is under attack by insects, the electrical signals can alert other parts of the network, triggering a defense response, perhaps by stimulating the production of chemicals that deter the insects. The network also seems to be involved in memory. Studies have shown that when a tree is damaged, the mycelial network retains information about the damage, even after the tree itself has died. This suggests that the network isn't just reacting to immediate threats, but is storing and processing information over longer periods. It's as if the forest is holding a collective memory, passed down through generations of fungal threads. Perhaps one of the most astonishing discoveries about the mycelial network is its role in facilitating the transfer of spores between trees. When a tree dies, the fungal network decomposes the wood, releasing vast quantities of spores. These spores then travel through the network, finding their way to other trees, helping them to regenerate and spread. It's a remarkable mechanism for forest regeneration, a quiet, almost imperceptible process that has been shaping our forests for millennia. The interconnectedness of the mycelial network creates a level of resilience within the forest ecosystem. When one area is damaged by fire, disease, or storm, the network can help to redistribute resources and support the recovery of the affected area. It's like a vast, underground support system, constantly working to maintain the health and stability of the forest. The scale of the mycelial network is truly staggering. In some old-growth forests, the network spans entire landscapes, connecting trees across vast distances. Scientists estimate that the total length of fungal hyphae in a single forest could be equivalent to several times the circumference of the Earth. It's a breathtaking testament to the power and complexity of nature, a hidden world teeming with life and intelligence. Researchers are continuing to unravel the mysteries of the mycelial network, exploring its potential applications in fields like medicine and bioremediation. The network's ability to transport nutrients and facilitate communication could be harnessed to deliver drugs to specific locations within the body, or to clean up polluted environments. But even without these potential applications, the mycelial network remains a profoundly beautiful and fascinating example of the interconnectedness of life on Earth. It's a reminder that the most important things aren't always the most visible, and that the deepest intelligence often resides beneath our feet. The forest floor breathes, not with the rise and fall of lungs, but with the slow, persistent expansion of a hidden architecture. It's a world built not of stone or wood, but of incredibly fine threads extending outward in every direction, a silent, tireless construction project orchestrated by the fungal kingdom. These threads, the hyphae, are the fundamental building blocks of what we're beginning to understand as fungal life, a vastly different and far more intricate system than anything we typically encounter in the plant or animal kingdoms. A single hypha, in its simplest form, is a remarkable structure. It's essentially a single, elongated cell, incredibly thin, often less than a human hair's width, and remarkably resilient. But don't let its delicate appearance fool you. These threads can grow to astonishing lengths, sometimes spanning hundreds of feet, reaching out across vast distances beneath the soil, within rotting logs, and even clinging to the roots of trees. This growth isn't driven by a central nervous system. Instead, it's a highly sensitive, exploratory process. The hyphae constantly probe their surroundings, seeking out nutrients and moisture. Specialized regions at the tip of each hypha, called growth points, are incredibly responsive to chemical signals, tiny molecules released by other fungi, decaying organic matter, or even by the plants themselves. These chemical signals act like whispers, guiding the hyphae towards sources of food. The hyphae are essentially equipped with sophisticated taste buds, capable of detecting sugars, amino acids, and other organic compounds. Some hyphae, particularly those of saprophytic fungi, the ones that feed on dead and decaying matter, are particularly adept at breaking down complex molecules. They possess powerful enzymes, biological catalysts that can dismantle cellulose, lignin, and chitin, the tough structural components of wood and insect exoskeletons. This remarkable ability allows them to recycle nutrients back into the ecosystem, playing a crucial role in decomposition and nutrient cycling. The beauty of the hyphae system lies in its branching structure. As a hypha encounters a promising source of nourishment, it doesn't simply consume it. Instead, it forks, dividing into two or more new hyphae, extending in different directions. This branching creates a vast, interconnected network, the mycelium, and it's this network that truly defines the fungal organism. It's like a tree, but instead of leaves and branches, it has countless interwoven hyphae. The density of the mycelium can vary dramatically depending on the species and the available resources. In nutrient-rich environments, the mycelium might be incredibly dense, forming a tightly packed mat. In leaner conditions, it becomes more sparse, with hyphae extending further and further in search of sustenance. Beyond simple feeding, the hyphae play a far more complex role in the forest's ecosystem. The interconnectedness of the mycelium isn't just a passive network, it's an active communication system. Through the release and detection of chemical signals, hyphae can transmit information about environmental changes, drought, flooding, or the arrival of herbivores to other parts of the network. This allows the fungi to coordinate their responses, influencing the behavior of plants and other organisms. Interestingly, scientists are discovering a symbiotic relationship between fungi and plant roots, particularly through mycorrhizal associations. The hyphae of certain fungi extend outwards, forming a network that wraps around the roots of trees and shrubs. In exchange for sugars produced by the plant through photosynthesis, the fungi provide the plant with essential nutrients like phosphorus and nitrogen, which are often scarce in the soil. This partnership is so critical that some scientists believe it's essential for the survival of many forest ecosystems. The fungal network essentially acts as an extension of the plant's root system, dramatically increasing its access to nutrients and water. The hyphae aren't just passively absorbing nutrients. They actively shape the soil itself. As they grow and die, they contribute to the formation of soil aggregates, improving soil structure and aeration. This, in turn, benefits plant growth and the overall health of the ecosystem. Furthermore, the hyphae can even influence the movement of water through the soil, creating pathways that facilitate the flow of water to plant roots. The sheer scale of the mycelial network is astounding. In some old-growth forests, the total length of fungal hyphae may exceed the circumference of the Earth several times over. It's a hidden continent beneath our feet, a silent, industrious world teeming with complex interactions. As researchers continue to investigate the secrets of the fungal kingdom, we're beginning to appreciate the profound and often overlooked role that these tiny threads play in shaping our planet's ecosystems. The fungal hyphae aren't just building blocks, they're the very foundation upon which many of the forest's wonders are built. The wind whispered through the jagged peaks of the Scottish Highlands, carrying with it the scent of damp earth and something subtly sweet, almost citrusy. This particular aroma wasn't produced by any single plant or tree. Instead, it emanated from a tapestry of color clinging to the rocks, a dazzling display of orange, yellow, green, and gray, collectively known as lichen. These weren't merely pretty patches on stone. They were remarkable examples of a truly ancient and astonishing partnership, a living testament to the incredible adaptability of the fungal kingdom. At the heart of this partnership lies the fungus, a crucial player we've been exploring. But what makes lichen so different, so resilient, is the addition of another vital element, algae or cyanobacteria. These photosynthetic organisms, remarkably simple in their cellular structure, are the engine of this symbiotic relationship, providing the energy that fuels the entire system. Think of it like this: the fungus provides the shelter, the structure, and crucially, access to water and minerals drawn from the rock. In return, the algae or cyanobacteria capture sunlight and transform it into sugars, feeding the fungus. The specific type of algae or cyanobacteria involved varies greatly depending on the environment. In colder, wetter climates like the Scottish Highlands, green algae, often Bryophyta species, are frequently dominant. These algae contribute to the characteristic green hues you see. In warmer, drier regions, you might find cyanobacteria, which are adept at surviving in harsher conditions and often produce vibrant yellows and oranges. The color shift isn't just for show. It's directly related to the specific photosynthetic pigments within the algal partner. The structure of a lichen is itself a beautiful illustration of this collaboration. What appears to be a single organism, the lichen you see on a rock, is, in reality, a tightly interwoven community. The fungal component, known as the mycelial, forms a dense, often branching network called a cortex. This cortex is incredibly strong, protecting the algal partner, the photobiont, from the harsh elements, intense sunlight, drying winds, and freezing temperatures. It's a remarkably sophisticated shield, built layer upon layer. Beneath the cortex lies the medulla, a spongy layer that acts like a water reservoir, keeping the photobiont hydrated even during extended periods of dryness. This is particularly important because the algae's photosynthetic machinery needs a constant supply of water to function efficiently. The fungal hyphae themselves, those incredibly fine threads we previously explored, are crucial for this water transport, channeling moisture from the surrounding environment directly to the algae. The arrangement isn't random. The algae cells are typically organized into rounded shapes called thalli, which are embedded within the fungal cortex. These thalli are arranged in a way that maximizes their exposure to sunlight, while simultaneously providing the fungus with optimal protection. It's a carefully orchestrated balance, a miniature ecosystem built around the needs of both partners. Interestingly, the lichen's ability to colonize such diverse surfaces, from rocks and tree bark to even human skin, demonstrates the remarkable tolerance of the fungal partner. Lichens can thrive in environments that would be completely hostile to the algae alone. The fungus has evolved to withstand extreme temperatures, high levels of radiation, and even the damaging effects of pollutants. This tolerance is due to the specialized adaptations within its hyphae, including the production of pigments that shield the algae from harmful UV rays. The longevity of lichens is truly astonishing. Some species have been found to be thousands, even tens of thousands of years old. This incredible lifespan is a direct result of the symbiotic relationship. The fungus provides a stable, sheltered environment, allowing the algae to continue photosynthesizing and producing energy. The algae, in turn, constantly renew the fungal component, effectively rebuilding the lichen from the ground up. It's a continuous cycle of growth, maintenance, and regeneration. Beyond their physical resilience, lichens play a vital role in their ecosystems. They are often the first organisms to colonize bare rock, initiating the process of soil formation. As the lichen dies and decomposes, it gradually breaks down the rock, releasing nutrients back into the environment. This process is critical for establishing the foundation for more complex plant life to eventually thrive. Furthermore, lichens contribute significantly to the carbon cycle, absorbing carbon dioxide from the atmosphere and storing it in their tissues. They act as a vital link in the transfer of carbon between the atmosphere and the ground. The very air we breathe is, in part, thanks to the efforts of these unassuming partnerships. The study of lichens continues to reveal new and fascinating insights into the complexities of symbiosis and the incredible adaptability of life on Earth. They represent a living bridge between the simplest photosynthetic organisms and the most complex fungal networks, demonstrating the power of cooperation and the beauty of interdependence. As the wind carried the sweet citrus scent across the highlands, one could almost hear the silent, ancient conversation between fungus and algae, a testament to a partnership that has endured for millennia. The air in the ancient forests of Gondwana, a time vastly different from our own, hung thick and humid. Ferns, towering in height, formed a dense, emerald canopy, punctuated by the burgeoning forms of early vascular plants, ancestors to the trees and flowers we know today. But something else was subtly shaping these landscapes, something far older and more persistent than the initial colonization by plants. These were the rust fungi, and their story is a remarkable one, a tale woven through billions of years of plant evolution. Rust fungi aren't flashy. They aren't the bright, colorful mushrooms that often capture our attention. Instead, they appear as often muted patches of orange, brown, or reddish-orange staining the leaves and stems of plants. At first glance, they might seem like a nuisance, a simple discoloration. However, scientists now understand that these stains represent one of the oldest and most pervasive forms of parasitic interaction on Earth, a relationship that played a critical, often overlooked, role in driving the diversification of plant life. The remarkable thing about rust fungi is their tenacity. They have persisted for over 400 million years, continuously evolving alongside the plants they infect. This longevity suggests a deeply ingrained and incredibly effective strategy. They don't simply cause cosmetic damage, they actively manipulate their hosts, forcing them to adapt and, in a crucial way, pushing them to become more complex. The initial interactions were likely less about devastating infection and more about a slow, gradual pressure. Early vascular plants, relatively new to the scene, possessed fewer defenses against pathogens compared to the more established mosses and liverworts. The rust fungi, arriving on the scene, began to exploit this vulnerability. They didn't initially aim to kill their hosts, but instead, they focused on extracting nutrients, particularly sugars, from the plant's tissues. This initial parasitic pressure, though seemingly minimal, created a selective environment. Plants that could tolerate, or even subtly resist, the fungal attack were more likely to survive and reproduce. Over countless generations, this selection pressure favored plants that developed enhanced defenses. These defenses weren't always elaborate immune systems. Sometimes, they were subtle changes in leaf structure, a slightly thicker cuticle, a different arrangement of cells that made it marginally harder for the fungi to penetrate. Other times, they involved the production of chemicals that deterred the fungi's growth. Crucially, the rust fungi didn't just sit back and watch. They responded to the defenses developed by their hosts, undergoing their own evolutionary adaptations. This created an ongoing evolutionary arms race, a dynamic feedback loop that shaped the trajectory of plant evolution. The fungi would evolve strategies to overcome a new defense, and the plants would, in turn, evolve new defenses. The diversity of rust fungi is astounding. There are over 13,000 different species, each specialized to infect a particular group of plants. Some rusts target ferns, while others specialize in gymnosperms like conifers, and still others infest flowering plants, including many economically important crops like wheat and rye. This incredibly broad range reflects their long history of interaction with a constantly changing botanical landscape. But perhaps the most fascinating aspect of rust fungi's influence is their role in the evolution of vascular tissue. Vascular plants, with their intricate system of xylem and phloem for transporting water and nutrients, are a defining characteristic of the plant kingdom. The prevailing theory suggests that rust fungal infections played a key role in the development and diversification of this vascular system. The initial stages of infection often involve the fungus penetrating the plant's cells, disrupting the normal flow of nutrients. As the fungus attempted to extract resources, it inadvertently triggered a strengthening of the plant's vascular tissues. The plant's response to the fungal attack, a localized thickening and reinforcement of the xylem and phloem, created a more robust and efficient system for transporting water and nutrients. Essentially, the rust fungi, in their relentless quest for sustenance, spurred the plants to develop a more sophisticated transport system. Furthermore, the repeated cycles of infection and healing, as the plant attempted to clear the fungus, contributed to the development of specialized cells and tissues, such as the cambium, responsible for producing new bark and wood that we see in mature trees. The very structures that give trees their strength and resilience were, in part, shaped by the persistent pressure of these ancient parasites. Looking at the fossil record, we can find evidence of this intricate relationship going back hundreds of millions of years. Microscopic analysis of fossil leaves reveals the tail-staining patterns of rust fungi, confirming their presence in ancient plant communities. It's a silent testament to a long and ongoing story. Today, rust fungi remain a vital part of forest ecosystems, contributing to nutrient cycling and facilitating the exchange of genetic material between plants. While they may not be celebrated for their beauty, they are undeniably significant players in the ongoing drama of plant evolution, a reminder that even the most seemingly destructive forces can, ultimately, contribute to the astonishing complexity and diversity of the plant kingdom. Their story is a complex, interwoven tale of survival, adaptation, and the continuous push and pull between parasite and host, a story that continues to unfold even now. Deep within the humid undergrowth of Southeast Asia, a silent drama unfolds, a captivating dance of survival and death orchestrated by a remarkable organism, cordyceps. These aren't your typical mushrooms growing peacefully on decaying wood. Cordyceps have a far more direct and, frankly, unsettling relationship with the animal kingdom, specifically insects. It's a story about a fungal invasion, a complete takeover, and a chilling demonstration of evolutionary adaptation. The most famous members of the Wars Cordyceps genus, particularly Cordyceps militaris and as Cordyceps floridula, are those that target caterpillars and beetles. But the story isn't limited to these. Several species now recognized exist, each with a specific preference, meticulously honed over millennia. It begins with the fungus identifying its host. Cordyceps spores, tiny and incredibly resilient, drift through the air, landing on a suitable host, often a vulnerable larva or pupa nestled within leaf litter or a decaying log. The spore, once landed, germinates, sending out a thread-like structure called a hypha. This hypha, like its brethren from other cordyceps, actively seeks out nutrients, and in this case, it aggressively targets the tissues of the insect. What sets cordyceps apart isn't just its parasitic nature, it's the astonishing precision of its attack. The hyphae don't simply feed. They systematically destroy the insect's nervous system. They release enzymes that break down the insect's muscles, nerves, and ultimately, the brain. This process isn't instantaneous, though. It's a slow, agonizing transformation. Initially, the host insect displays typical larval behaviors, crawling, feeding, and attempting to pupate. However, as the cordyceps gains control, the insect becomes increasingly immobile, its movements jerky and uncontrolled. It might attempt to burrow deeper into the soil, or frantically thrash, a desperate, futile struggle against a force it doesn't understand. The beauty, and the horror, of this process lies in the fungal manipulation of the host's behavior. Cordyceps doesn't just kill, it redirects. The exact mechanism of this behavioral control is still being researched, but scientists believe it involves the release of specific chemical compounds that interfere with the insect's neurotransmitters, the chemicals responsible for transmitting signals in the nervous system. These compounds essentially hijack the insect's control center, compelling it to perform actions that benefit the fungus. Once the host is effectively paralyzed, cordyceps begins to grow a stalk, a fruiting body directly from the insect's body. This stalk, reaching upwards, emerges above the soil, often positioned perfectly to allow the spores to disperse into the wind. The fungal mass at the top, known as the shroom, is a stunning sight of vibrant, often fluorescent, display of color, a stark contrast to the decaying insect beneath. The color of the shroom, including shades of pink, orange, and purple, isn't just for show, it's a signal, attracting insects and other arthropods that carry the spores to new hosts. The relationship between cordyceps and its host is a remarkable example of co-evolution. The insect larvae have evolved defense mechanisms to avoid a cordyceps, such as choosing hosts with lower fungal densities or developing thicker exoskeletons. But cordyceps has continually adapted, evolving more potent toxins and strategies to overcome these defenses. It's a perpetual game of cat and mouse, a testament to the relentless pressures of natural selection. Interestingly, the fungal infection isn't always a complete takeover. Sometimes, the S. cordyceps infection is partially successful, resulting in a host that is weakened and exhibiting altered behavior, but still capable of some movement and feeding. These partially infected individuals offer a valuable window into the intricate dynamics of this deadly interaction. Beyond caterpillars, a cordyceps also targets beetles. The beetle's influence on the fungal growth is particularly fascinating. The beetle's burrowing activities, inadvertently created by the fungus, expose the insect's larvae to a larger area of soil, increasing the likelihood of spore dispersal. The beetle, in a strange twist, effectively acts as a dispersal agent, contributing to the fungus's reproductive success. The study of cordyceps isn't just an academic exercise. Researchers are investigating the antifungal compounds produced by cordyceps, which have shown promise in treating human fungal infections. The potent toxins within these fungi could potentially lead to new antibiotics, offering a crucial weapon against drug-resistant pathogens. Furthermore, understanding the mechanisms behind a cordyceps behavioral manipulation could have implications for fields like neuroscience and behavioral psychology. As the sun sets over the humid forest floor, casting long shadows through the undergrowth, the silent drama of the cordyceps continues. A tiny shroom rises above the decaying insect, a symbol of the complex, often brutal, beauty of the natural world, and a reminder of the extraordinary power of fungal life. The forest floor isn't just dirt and fallen leaves. It's a bustling, hidden world, a slow-motion symphony of decay orchestrated by a kingdom largely unseen, the wood decay fungi. These aren't the mushrooms you might find popping up after a rainy day, though many are, but rather a vast, interconnected network of organisms quietly, persistently transforming the very essence of trees. They are, in effect, the architects of decomposition, silently and diligently rebuilding the world in a way entirely their own. The process begins with the death of a tree. It's a natural event, a return to the earth, but without the fungi. That return would be far slower, far more disruptive. Initially, bacteria, driven by their appetite for readily available sugars and starches, begin to break down the outer layers of the wood. However, wood is remarkably resilient. Its complex structure, composed primarily of cellulose and lignin, substances notoriously difficult for most organisms to digest, provides a formidable defense. This is where the wood decay fungi step in, deploying a suite of remarkable biochemical weapons. These fungi aren't uniform. There are many different types, each specializing in different stages and aspects of wood decomposition. Some, like the bracket fungi, those shelf-like growths you occasionally see clinging to trees, are pioneers, rapidly colonizing the fresh, moist surface of a fallen log. They are often brightly colored, a dazzling display of pigments produced for protection and to attract insects that aid in spore dispersal. The colors aren't just aesthetic. Many contain compounds that deter herbivores or act as potent antibiotics, safeguarding the fungal colony. These early colonizers specialize in breaking down the softer, more accessible parts of the wood, releasing simple sugars and other compounds. As the initial assault subsides, more specialized fungi arrive. White rot fungi, characterized by their ability to degrade lignin, the glue that holds wood together, are particularly important. They produce powerful enzymes, like lignin peroxidase, that essentially dissolve the lignin, leaving behind a bleached, porous structure. This type of decay, resulting in a pale, spongy wood, is often associated with fungi like Inonotus obliquus, the shiitake mushroom, though many species are less recognizable. The breakdown of lignin opens up the wood further, allowing other fungi to access the cellulose. Brown, rot fungi, on the other hand, primarily target the cellulose itself, leaving behind a darkened, crumbly wood. They don't typically attack the lignin, focusing instead on the carbohydrate components. Species like a Tremetes versicolor, commonly known as turkey tail, are masters of this process. Their activity contributes significantly to the formation of humus, the dark, rich organic matter that improves soil quality. But the influence of wood decay fungi extends far beyond simply breaking down wood. Their metabolic processes are intrinsically linked to the global nutrient cycle. As they dismantle wood, they release a constant stream of essential nutrients, nitrogen, phosphorus, potassium, back into the soil. These nutrients are then readily available for plant uptake, fueling the growth of new trees and other vegetation. It's a beautifully closed loop, a testament to the intricate interconnectedness of life. Moreover, these fungi play a crucial role in carbon sequestration. As they break down complex carbon compounds in wood, they release carbon dioxide back into the atmosphere. However, the carbon is then incorporated into the soil as humus, effectively removing it from the active cycle for long periods. The fungal networks themselves are astonishingly complex. Many wood decay fungi form symbiotic relationships with tree roots, exchanging nutrients and signaling compounds. This mycorrhizal network, a vast web of fungal threads extending through the soil, dramatically increases the efficiency with which trees absorb water and nutrients, while simultaneously providing the fungi with carbohydrates produced through photosynthesis. This mutualistic relationship is fundamental to the health and resilience of most forests. Furthermore, the diversity of wood decay fungi contributes to the stability of forest ecosystems. Different species specialize in degrading different wood types, helping to maintain a balance and preventing the dominance of any single species. They are, in essence, a control mechanism, constantly adjusting to changing conditions and ensuring that wood remains a source of nutrients rather than a barrier to new life. The process isn't just about the immediate decomposition of a fallen tree. The accumulated humus created by wood decay fungi over centuries, millennia, even longer, forms the foundation of many soils, influencing their texture, fertility, and drainage properties. These soils, in turn, support a vast array of other organisms, including insects, worms, and other microbes, creating a thriving ecosystem. It's a cascading effect, a chain reaction initiated by the silent work of these fungal architects. Even the rate at which wood decays is influenced by the fungi. Different fungal communities create different decay patterns, shaping the landscape in unique ways. The patterns of decay across a forest floor are like a fungal fingerprint, revealing information about the history of the ecosystem, the age of the trees, and the prevailing environmental conditions. As the forest continues to cycle, the influence of wood decay fungi remains constant, a slow, persistent force shaping the world around us. They are a testament to the power of microbial communities and their critical role in sustaining life on Earth, a hidden kingdom diligently working to transform, rebuild, and nourish the planet. The forest floor, already a tapestry of decaying leaves and damp earth, held a secret even more astonishing than the intricate networks of mycelium revealed in the previous parts of our exploration of world of fungi. It wasn't a secret whispered by the rust's patient assault on plant tissue, nor a dark manipulation by the cordyceps, nor the slow, methodical dismantling of wood by dedicated decomposers. This was a light, a soft, ethereal glow emanating from the very fabric of the forest, a phenomenon that baffled and delighted researchers for decades. Fungal bioluminescence. The story of this living light began, surprisingly, in Japan. In 1961, a Japanese mycologist named Dr. Shinya Kumagai was studying Panis eucalyptus, a small, unassuming mushroom known locally as lantern mushroom. He was meticulously examining specimens in his laboratory when he noticed something extraordinary. Under complete darkness, the gills of the mushroom were emitting a faint, greenish, yellow light. Initially, he suspected a contamination, some phosphorescent material clinging to the mushroom's surface. However, rigorous testing revealed that the light was intrinsically part of the fungus itself. It wasn't reflected or produced by external chemicals. It was generated by the fungus's own biological processes. The mechanism behind this living light is still not entirely understood, but scientists have made significant progress. The most common understanding centers around a molecule called luciferin, a light-emitting compound, and an enzyme called luciferase. Luciferase acts as a catalyst, speeding up a chemical reaction involving luciferin. When luciferin reacts with oxygen, water, and ions like magnesium, it produces an excited molecule. This excited molecule quickly returns to its normal state, releasing energy in the form of light. Interestingly, the color of the light produced varies considerably depending on the fungal species. Panis eucalyptus, the original discoverer of this incredible phenomenon, emits a characteristic yellow-green light. But other bioluminescent fungi, like Mycena luxeaterna, found in the rainforests of Vietnam, produce a vibrant blue light. The variation in color suggests that different fungal species utilize different luciferin-luciferase systems, each optimized for a particular wavelength of light. The purpose of this bioluminescence remains one of the great mysteries of fungal biology. Numerous theories have been proposed, and research continues to explore each one. One prevalent hypothesis suggests that the light attracts insects, primarily beetles, which then aid in spore dispersal. The insects, drawn to the glow, land on the mushroom gills, picking up spores and carrying them to new locations. This is supported by the observation that many bioluminescent fungi grow in areas with high insect activity. However, not all bioluminescent fungi have a strong association with insect activity, suggesting that the light may serve other functions. Another theory posits that the light serves as a warning signal to deter herbivores. A bright, unusual glow could make a mushroom appear unpalatable or even toxic, discouraging animals from consuming it. This is a common strategy employed by other organisms, and it's plausible that bioluminescence evolved as a defensive mechanism in fungi. Furthermore, scientists believe that bioluminescence might play a role in attracting mates. In fungi, reproduction often involves the exchange of genetic material between hyphae, the thread-like filaments that make up the fungal network. Bioluminescence could act as a visual signal, guiding hyphae from different individuals to one another, facilitating this crucial reproductive process. This is particularly intriguing considering that some bioluminescent fungi exhibit synchronized flashing, as if communicating in a silent, pulsing language. The distribution of bioluminescent fungi across the globe offers a remarkable snapshot of evolutionary adaptation. They are found in tropical and subtropical regions, primarily in areas with high humidity and abundant, decaying organic matter, the ideal conditions for fungal growth. However, recent discoveries have revealed bioluminescent fungi in more temperate climates as well, suggesting that this trait may be more widespread than previously thought. The presence of bioluminescent fungi in caves, for instance, points to a potential role in adapting to the darkness. The study of fungal bioluminescence has implications far beyond simply documenting a beautiful natural phenomenon. It could potentially lead to new technologies based on the principles of bioluminescence. Researchers are exploring the possibility of using fungal luciferase to develop highly sensitive biosensors, capable of detecting even minute traces of chemicals or pollutants. The stability and efficiency of fungal luciferase make it an ideal candidate for such applications. Moreover, understanding the mechanisms behind fungal bioluminescence could unlock new insights into the evolution of light production in other organisms, including plants and animals. It's a reminder that the natural world is full of surprises, constantly revealing new wonders and challenging our understanding of life itself. As we continue to explore the depths of fungal ecosystems, we are likely to uncover even more extraordinary secrets, shining brightly in the darkness. The scent of warm yeast and rising dough hangs heavy in the air, a surprisingly ancient aroma that has shaped human civilization for millennia. It's a scent inextricably linked to fermentation, and at its heart lies the magic of fungi. While we often think of bacteria as the primary agents in turning sugary substances into delicious and complex flavors, the vast majority of traditional fermentation processes, from the simple act of baking bread to the intricate art of brewing beer, rely almost entirely on the remarkable capabilities of yeast, which, remarkably, are fungal in origin. The story of yeast's influence begins not in a brewery or bakery, but in the incredibly intricate ecosystems of decaying wood. The fungi we explored in Part 6, those tireless decomposers, were breaking down cellulose, a remarkably strong and complex carbohydrate found in plant cell walls. As they did this, they produced ethanol, a simple alcohol, as a byproduct. This isn't a deliberate, targeted action, rather. It's a consequence of their metabolic process. For hundreds, perhaps thousands of years, tiny, airborne yeasts, initially carried on the wind and deposited in warm, moist environments, began to colonize these fungal mats. These weren't the yeasts we find in commercial brewing today. They were wild yeasts, perfectly adapted to the conditions of rotting wood. Early humans, lacking sophisticated understanding of microbiology, stumbled upon a remarkable observation when they stored fruits, grains, or honey in porous containers, think clay pots or woven baskets. These wild yeasts would actively ferment the sugars within, transforming them into alcoholic beverages. The Greeks, long before Aristotle, recognized this phenomenon, referring to it as zethus, which translates roughly to fermentation. They used it for medicinal purposes, believing fermented honey possessed healing properties. The Romans, inspired by the Greeks, also utilized fermented foods and drinks, although their knowledge remained largely practical rather than scientific. As societies developed, so did the techniques of controlled fermentation. The ancient Egyptians, famous for their advanced knowledge of agriculture and engineering, are credited with developing the first true bread-making techniques. They utilized a strain of Saccharomyces cerevisiae, a yeast species we still use today, to transform flour, water, and a small amount of levain, a pre-fermented dough, into the flat, dense loaves that formed the staple of their diet. The key wasn't just the yeast itself, but the careful maintenance of a starter, a continuously refreshed culture of yeast and bacteria, which provided a consistent and predictable fermentation environment. This starter, passed down through generations, became a vital part of their culinary heritage. The practice of brewing beer followed a similar trajectory. Initially, brewers relied on whatever wild yeasts happened to be present in the grains they used. However, over time, as brewing became a more refined art, brewers began to actively select and cultivate yeasts with specific characteristics. Saccharomyces cerevisiae remained a dominant player, but different strains developed over centuries, each contributing subtly different flavors and aromas to the final product. Early beer was a far cry from the highly carbonated lagers we often enjoy today. It was typically dark, strong, and fermented at warmer temperatures, relying heavily on the wild yeasts for flavor and body. The scientific understanding of fermentation didn't emerge until the 19th century, thanks to the pioneering work of scientists like Louis Pasteur. Pasteur's meticulous experiments demonstrated that fermentation was not a spontaneous process, but rather driven by living organisms, specifically yeast consuming sugars and producing alcohol and carbon dioxide. He identified Saccharomyces cerevisiae as the primary agent responsible and developed methods for controlling fermentation, effectively laying the foundation for modern brewing and baking. Today, the fermentation industry is a global powerhouse, encompassing everything from artisanal craft breweries to massive industrial food production. Modern yeast strains have been meticulously selected and engineered to produce specific flavors and aromas, allowing brewers and bakers to create an astonishingly diverse range of products. From the delicate floral notes of a Belgian ale to the robust flavors of a sourdough loaf, each step relies on the ongoing metabolic activity of Saccharomyces cerevisiae and other yeast species. It's a beautiful example of the profound and enduring influence of fungi, a constant reminder that even the simplest of human pleasures are deeply rooted in the intricate workings of the natural world. The warmth of the oven, the satisfying fizz of a freshly opened bottle, these are all echoes of an ancient process, a testament to the silent, tireless work of these remarkable fungal allies. The rain in Slime Valley had ceased, leaving behind a quiet, shimmering surface. The air, thick with the scent of damp earth and decaying leaves, carried a subtle, almost metallic tang, a consequence of the intricate chemical reactions constantly taking place beneath the forest floor. This particular scent, though initially unnoticed, would soon become central to one of the greatest breakthroughs in medical history, a story intrinsically linked to a humble, unassuming fungus. It's a story about a mold, Apis penicillium notatum, and how it inadvertently saved countless human lives. For centuries, bacterial infections were a terrifying threat. A simple cut, a scratch, or even a minor wound could quickly escalate into a deadly fever. Doctors had limited understanding of what caused these illnesses and, even worse, few tools to combat them. Surgical procedures were often hampered by the risk of infection, and mortality rates from common illnesses were staggeringly high. The prevailing wisdom held that infection was simply the body's reaction to an evil, invading force, a concept that offered little in the way of treatment. Then, in 1928, a young Scottish bacteriologist named Alexander Fleming was examining a collection of Staphylococcus bacteria he'd grown on agar plates. He was a meticulous researcher, dedicated to understanding the invisible world of microorganisms. Fleming, working at St. Mary's Hospital in London, wasn't seeking a cure for bacterial infections, but rather investigating how bacteria behaved. He was studying ways to prevent infections in wounds. A pressing concern during World War I, he noticed something peculiar. One of his agar plates, intended to house a pure culture of Staphylococcus, had been contaminated. A fuzzy, blue-green mold had sprouted, and surrounding the mold's growth were clear zones, areas where the pure Staphylococcus bacteria simply hadn't grown. It was as if the mold was actively preventing the bacteria from multiplying. Fleming, a keen observer, immediately recognized the significance of this observation. He carefully identified the mold as Penicillium notatum, a species previously considered harmless and largely ignored. The blue-green pigment produced by the mold, called penicillin, was the key. Penicillin, he discovered, was producing a substance that inhibited the growth of bacteria by interfering with their ability to build cell walls. Bacteria require these rigid walls to survive, and penicillin effectively disrupted this process, essentially starving the bacteria. Fleming published his findings in 1929, detailing the antibacterial properties of penicillin. However, the scientific community, at the time, wasn't immediately receptive. The research was published in the British Journal of Experimental Pathology, a relatively obscure journal, and the initial response was limited. The mechanisms behind penicillin's action weren't fully understood, and the sheer novelty of the discovery was somewhat overlooked. Further attempts to isolate and purify penicillin were unsuccessful, hampered by the mold's instability and the difficulty in extracting the active compound. Years passed. Fleming continued his research, meticulously documenting his observations and experimenting with different techniques. He even used penicillin successfully on mice, demonstrating its effectiveness against several bacterial infections. Yet, the scientific world remained largely unconvinced. It wasn't until the late 1930s that Howard Florey and Ernst Chain, researchers at Oxford University, took up Fleming's work. They, along with their team, were captivated by the potential of penicillin. Florey and Chain systematically investigated Fleming's findings, developing methods to isolate and purify penicillin in larger quantities. They faced immense technical challenges, struggling to produce enough of the substance for meaningful testing. Their persistence, coupled with innovative techniques, eventually led to a breakthrough. In 1941, they successfully produced enough penicillin to conduct the first clinical trials on humans. The results were astonishing. A seriously ill policeman, Mr. Albert Charles Dennis, suffering from a severe and rapidly progressing bacterial infection, received penicillin as an experimental treatment. Within days, Dennis's condition dramatically improved. The infection, which had been unresponsive to conventional treatments, began to subside. This was a pivotal moment, providing undeniable proof that penicillin could indeed be effective in treating human diseases. News of these early successes spread rapidly. With the onset of World War II, the urgent need for effective antibacterial treatments became even more pressing. The demand for penicillin soared, and Florey and Chain, with the assistance of the U.S. pharmaceutical company Merck, established large-scale production facilities. By 1945, penicillin was widely available, transforming the treatment of bacterial infections and saving countless lives during the war and beyond. The discovery of penicillin, originating from a seemingly insignificant mold found in a forgotten corner of Slime Valley, fundamentally changed the landscape of medicine. It demonstrated the profound potential hidden within the fungal kingdom, a kingdom previously largely dismissed as agents of decay. S. Penicillium notatum became a symbol of scientific serendipity, a reminder that groundbreaking discoveries can arise from unexpected observations and persistent investigation. The story of penicillin is a testament to the intricate connections within the natural world, and to the remarkable capacity of science to harness those connections for the benefit of humanity. The faint, metallic scent in Slime Valley continued, now associated not with decay, but with a miracle. The ocean floor, far beneath the dancing sunlight, is a place of incredible pressure and, surprisingly, burgeoning life. For decades, scientists believed coral reefs were solely the domain of brightly colored fish, intricate invertebrates, and the calcium carbonate skeletons of the coral themselves. But recent expeditions, fueled by the detection of unusual electrochemical signals, have revealed something astonishing: a vast, interconnected ecosystem built, in large part, by fungi. We call it the myco reef. The initial discovery came from Dr. Eris Thorne and his team at the Oceanic Research Institute. Thorne, a mycologist specializing in extremophile fungi, those that thrive in harsh environments, had been tracking subtle shifts in electrical conductivity around the Great Barrier Reef. The patterns were too complex to be explained by simple geological activity. They pulsed with a rhythm. using advanced submersible drones equipped with specialized biosensors. They began to map the source of these signals, and what they found was unlike anything they'd ever encountered. Vast networks of filamentous fungi, resembling glowing, branching coral in their own right, carpeted large sections of the reef floor, primarily in the twilight zone where sunlight barely penetrates. These weren't the familiar mushrooms we see on land. These fungi, dubbed luminospora by the team, possessed remarkable adaptations. Their hyphae, the thread-like filaments that make up the fungal body, were incredibly resilient, capable of withstanding the crushing pressure of the deep ocean. More strikingly, they exhibited a complex metabolic process that utilized dissolved organic matter, primarily the marine snow, the constant shower of decaying organic material sinking from the surface, as its primary energy source. But it wasn't just simple decomposition. The luminospora were actively processing and transforming this material, creating a network of chemical pathways that fueled not just themselves, but also a surprising range of other organisms. What's particularly fascinating is the symbiotic relationship that has developed. The luminospora aren't simply existing, they are facilitating a miniature food web. The fungi release compounds that encourage the growth of bacterial colonies, which in turn consume and break down the remains of dead marine life. These bacteria, in turn, are consumed by small crustaceans and worms that live within the fungal networks. Larger organisms, like certain species of sea snails and small, bioluminescent fish, have even been observed grazing directly on the fungal colonies, utilizing the nutrients released during the fungal metabolism. The team's research indicates the fungi play a crucial role in nutrient cycling within the myco-reef. They essentially act as biological transformers, converting organic waste into forms accessible to a multitude of organisms. This is amplified by the fungi's ability to sequester heavy metals, a common problem in polluted coastal waters, effectively detoxifying the environment around the reef. Some of the luminospora species possess specialized enzymes that bind to mercury and lead, preventing them from accumulating in other marine organisms and protecting the overall health of the reef. Beyond the direct feeding relationships, the electrical signaling itself is proving to be incredibly complex. Scientists believe the luminospora are communicating not with sounds, but with electrical impulses. These signals appear to be related to nutrient availability, predator avoidance, and even reproductive strategies. It's hypothesized that groups of luminospora can coordinate their metabolic activity, creating pulses of energy that attract other organisms to a particular location, essentially acting as a biological beacon. The team is currently investigating whether these signals could even be used for sophisticated information transfer, a primitive form of fungal language. Furthermore, the myco-reef exhibits a stunning display of bioluminescence far exceeding that of the typical coral reefs. While coral themselves produce some light through symbiotic algae, the luminospora's bioluminescence is significantly brighter and more varied. Different species produce light at different wavelengths, blues, greens, and even faint reds, and this light is believed to be used for a variety of purposes. Some researchers propose that the light attracts prey for predatory fish, while others suggest it's a form of camouflage, disrupting the outline of the fungal colonies against the dim background light. It's a dazzling spectacle, a silent, electric dance within the depths. The discovery of the myco-reef dramatically alters our understanding of marine ecosystems. It highlights the astonishing adaptability of life and demonstrates the vital role fungi play in shaping the world around us. The sheer scale of the network, spanning hundreds of square kilometers, suggests that similar myco-reefs may exist in other deep sea environments globally, waiting to be discovered. The research is still in its early stages, and many questions remain. For example, scientists are attempting to understand the full extent of the Luminospora's genetic diversity and how this diversity contributes to the resilience of the myco reef. The possibility of harnessing the fungi's unique properties, their ability to detoxify pollutants, their bioluminescence, or their nutrient cycling capabilities, is also being explored. As exploration continues, it's becoming increasingly clear that the ocean's depths are not just a vast, empty space, but a vibrant, interconnected world teeming with life, a world profoundly shaped by the silent, persistent work of fungi. It's a reminder that the most surprising discoveries often lie hidden beneath the surface, waiting to be unveiled by curious minds and advanced technology. The twilight deepened in the shadowed valleys of mycelia, the vast subterranean world of fungi. A gentle, rhythmic pulsing resonated through the network, a chorus of metabolic activity sustained by the constant exchange of nutrients and information. Tonight, the focus wasn't on the delicate processes of decomposition or the immediate gratification of feeding, but on something far more ancient and remarkable: the evolution of fungal armor. For millennia, these silent architects of life hadn't simply relied on their networks and chemical signals. They had crafted their own defenses, ingenious strategies for survival against a world that often sought to consume them. The key to this armor lay within the spore. Initially, fungal spores were remarkably simple, tiny, delicate structures designed primarily for dispersal. They floated on air currents, splashed through rainwater, or were carried by animals, seeking a suitable patch of substrate to germinate and establish a new mycelial colony. However, the environment posed significant challenges. Harsh sunlight could desiccate them instantly, drying them into brittle fragments. Mechanical abrasion from being bumped against rocks or trampled by larger organisms threatened to rupture their fragile bodies. And, perhaps most critically, predation existed. While larger organisms rarely considered fungi as a direct food source, smaller invertebrates and even certain microbes saw them as a convenient meal. Over vast stretches of time, the selective pressures of this hostile world triggered a series of astonishing adaptations. The earliest versions of what we now recognize as armor weren't elaborate shells or hardened exteriors. Instead, they were subtle changes in spore morphology, prioritizing resistance to desiccation and physical impact. Some species developed spore walls with significantly increased thicknesses, built from modified polysaccharides, complex sugars, that formed the very backbone of their structures. These walls weren't rigid, they were remarkably flexible, allowing the spores to bend and absorb the force of an impact, rather than shattering upon collision. Further refinements involved the incorporation of waxy coatings. Certain genera, particularly those inhabiting drier environments, produced spores with substantial layers of cutin, a polymer also found in plant leaves and insect exoskeletons. This cutin layer acted like a miniature waterproof suit, dramatically reducing water loss and increasing the spore's resilience against dehydration. The process wasn't simply about building up layers. The fungi developed specialized cells within the spore wall dedicated to synthesizing and regulating the production of these protective compounds, a complex biochemical factory operating within the tiny structure. But the most profound developments occurred with the evolution of what scientists now term mucilage armor. Some fungal spores, particularly those found in exceptionally challenging environments like volcanic vents or areas with high levels of radiation, began producing a thick, gelatinous substance, a complex mixture of sugars, proteins, and nucleic acids that encased the entire spore. This mucilage layer wasn't just a protective shield, it also provided a vital source of nutrients, allowing the spore to survive extended periods without a suitable substrate. It acted as a miniature, self-contained ecosystem, waiting for conditions to become favorable. The control of this mucilage production was astonishingly precise, often triggered by environmental cues such as changes in temperature, humidity, or the presence of certain chemicals. Some fungal species could even detect the presence of potential predators and immediately ramp up mucilage production, creating an almost impenetrable barrier around their spores. Researchers have identified specific enzymes within these spores that respond to mechanical stress, releasing compounds that reinforce the mucilage layer. It's a living response, a tiny, self-aware system constantly assessing and adapting. The incredible diversity of fungal armor is still being uncovered. Some species develop spores that resemble tiny pebbles, minimizing their visibility to potential grazers. Others produce spores that are incredibly small and lightweight, allowing them to be carried over long distances by even the gentlest breezes. The evolution of trigger spores, spores that germinate only when subjected to a specific shock or vibration, represents another extraordinary adaptation, likely a response to seismic activity or the movement of large animals. The intricate mechanisms underlying fungal armor are not merely about physical defense. They represent a sophisticated understanding of the environment, a sensing capability that allowed the fungi to predict threats and proactively prepare. It highlights a fundamental principle of evolution, the ability to turn a simple function dispersal into a robust strategy for survival. As scientists continue to delve deeper into the hidden world of mycelia, they are increasingly realizing that the fungal armor is not just a defense. It's a testament to the ingenuity and resilience of one of the most ancient and pervasive forces on Earth. It's a living demonstration of how, over eons, life adapts, innovates, and ultimately endures. Deep beneath the forest floor, in a realm largely unseen by human eyes, exists a complex and astonishing network. It's not made of wires or cables, but of thread-like structures, delicate and remarkably strong, weaving together the roots of countless trees and plants. This intricate system, known as the Wood Wide Web, is a silent conversation happening constantly, a vast and surprisingly sophisticated exchange of information and resources between the members of a forest community. Scientists have discovered that this remarkable network is built by mycorrhizal fungi, the most abundant organisms on Earth, quietly shaping the ecosystems around us. These fungi don't simply live alongside plant roots, they form symbiotic relationships, a partnership of mutual benefit. The plant provides the fungi with sugars, produced through photosynthesis, a tasty reward for the fungus's efforts. In return, the fungal network extends outwards, vastly increasing the root system's reach. Instead of being limited to the immediate vicinity of the plant's roots, the fungal hyphae, the fine thread, like filaments that make up the fungal body, can spread for meters, sometimes even tens of meters, seeking out water, nutrients like phosphorus and nitrogen, and minerals locked away in the soil. The soil itself can be a surprisingly poor provider of these elements, especially for the more demanding needs of large trees. It's like the fungi are acting as incredibly efficient, underground delivery services, bringing nourishment directly to the roots. But the Wood Wide Web is much more than just a transportation system. Researchers have begun to unravel a stunning aspect of this network-plant communication. It turns out that trees aren't solitary beings, focused only on their own survival. They can talk to each other through the fungal network, sending signals and receiving information, altering their behavior based on what they learn. This communication happens through chemical signals. When a tree is attacked by insects, for example, it can release volatile compounds into the air. These chemicals, carried by the wind, eventually reach the fungal network. The fungi, acting as scouts, recognize the threat and quickly transmit a warning signal back to neighboring trees. These receiving trees then begin to strengthen their defenses, producing chemicals that make their leaves less palatable to the insects, or even synthesizing defensive compounds that directly harm the attackers. It's a remarkably rapid response, often happening within hours. The fungal network doesn't just facilitate defense. It's also involved in the distribution of resources between trees of different ages and health. Older, larger trees, often referred to as mother trees, can invest energy and resources into younger saplings, providing them with crucial nutrients through the network. These mother trees, recognized for their resilience and vitality, can sometimes identify stressed or weakened saplings and focus their support on the ones most in need. This isn't altruism in the human sense, but a natural consequence of the interconnectedness of the wood wide web. A healthy, thriving mother tree benefits the entire forest community. Interestingly, the network isn't just connecting trees of the same species. Research has shown that plants of different species can also communicate through it. A young beech tree, for instance, has been observed to receive signals from a neighboring oak tree, influencing its growth patterns and even its susceptibility to disease. Scientists are still working to understand the full extent of this inter-species communication, but it's clear that the wood wide web is a surprisingly diverse and inclusive network. The fungal hyphae themselves aren't just passive conduits. They contain specialized cells that can transmit electrical signals, similar to neurons in animal brains, although far simpler. These electrical signals are thought to play a role in rapid communication within the network, allowing information to be transmitted much faster than through chemical signals alone. It's a truly remarkable system, demonstrating a level of complexity that researchers are only beginning to comprehend. The structure of the wood wide web also plays a significant role in its function. The network isn't a uniform, interconnected blob. Instead, it's composed of distinct highways, thicker, more densely packed areas of fungal hyphae that carry information more efficiently. These highways act as major communication routes, allowing for rapid dissemination of information throughout the forest. Furthermore, the network isn't static. It's constantly being remodeled and reshaped by the growth and decay of fungal hyphae. As fungi die and new ones grow, the network changes, adapting to the needs of the surrounding plants. This dynamic nature of the wood wide web is crucial to its resilience and its ability to sustain the forest ecosystem. The discovery of the wood wide web has fundamentally altered our understanding of forest ecology. It highlights the importance of considering the interconnectedness of all living things and demonstrates that forests are not simply collections of individual trees, but complex, collaborative communities. Studying this hidden network is not just about understanding plant communication, it's about recognizing the vital role that fungi play in maintaining the health and stability of our planet's most important ecosystems. As scientists continue to explore this astonishing realm, we can anticipate even more profound revelations about the secrets held within the wood wide web. The damp earth smelled particularly potent that autumn, a blend of decaying leaves and something subtly different, something sharper. For generations, indigenous peoples of the Amazon rainforest had observed an unsettling pattern amongst the local monkey populations. During the dry season, when fruit was scarce and the monkeys were desperately searching for sustenance, a strange lethargy would descend. They'd become docile, slow to react, and remarkably willing to approach human settlements, a behavior entirely unlike their usual cautious nature. Researchers began to suspect a connection, a subtle influence operating from the forest floor. The key, they realized, lay in a group of fungi previously considered merely decomposers, the Cordyceps species. Initially, the understanding of Cordyceps was rooted in observation and folklore. The tribes, particularly the Yanomami, possessed detailed knowledge of these fungi, passed down through oral traditions. They weren't seen as malevolent spirits, but rather as powerful agents, acting upon the will of the forest. Descriptions focused on the remarkable way the fungi would colonize insect hosts, beetles, grasshoppers, even crickets, growing directly out of the animal's body, often erupting from the head, before releasing spores. This dramatic transformation was interpreted as a control exerted over the host, a forceful direction given to its movements. While early European explorers dismissed these accounts as superstition, the sheer volume and consistency of the observations began to pique the interest of dedicated entomologists and mycologists. The first rigorous scientific investigations into cordyceps neurotoxicity began in the 1950s, spearheaded by a brilliant, yet somewhat eccentric, mycologist named Albert Pilarski. Pilarski, stationed in Brazil, was fascinated by the remarkable potency of Cordyceps militaris, initially discovered by Linnaeus in 1753. He meticulously documented the fungus's ability to induce paralysis in insects. He realized that the fungal mycelium, the thread-like network that forms the body of the fungus, didn't simply consume the host's tissues. It actively produced compounds, toxins, that interfered with the insect's nervous system. These compounds, collectively known as cordyceps toxins, were particularly effective at disrupting the transmission of nerve impulses, causing paralysis and ultimately death. What made Pilarski's work truly groundbreaking was his ability to isolate and identify several of these toxins. He discovered, for example, cordyceopin, a steroid-like compound that directly blocked the sodium channels in nerve cells, a critical component of nerve impulse transmission. He later identified other compounds, including cordycepin A and B, each exhibiting slightly different effects on the nervous system. His research demonstrated unequivocally that cordyceps was not simply a parasite, it was a sophisticated neurotoxin producer, utilizing a targeted mechanism to control its host's behavior. The understanding of this mechanism expanded significantly in the late 20th and early 21st centuries with advancements in biochemistry and molecular biology. Researchers began to unravel the specific pathways affected by the toxins, discovering that cordyceps could manipulate a range of neurological processes, including dopamine signaling, serotonin regulation, and even the expression of genes involved in muscle control. It wasn't just about stopping the nerves from firing. The fungus seemed to be actively reshaping the insect's brain, altering its priorities and directing its movements. Interestingly, the research has expanded beyond insects. Studies have shown that cordyceps can produce toxins that affect the behavior of small mammals, including rodents. The exact mechanisms are still being investigated, but it appears that the fungal toxins can cross the blood-brain barrier, reaching the central nervous system and influencing brain activity. The potential for manipulating animal behavior, even unintentionally, through fungal toxins became a significant area of research, particularly regarding agricultural pests. However, the implications extend further than simply controlling unwanted insects. The discovery of cordyceps' ability to influence movement has sparked interest in neurological disorders and even potentially in human health. Certain cordyceps compounds, notably cordycepin, are currently being investigated for their potential therapeutic properties, including their ability to stimulate the production of adenosine triphosphate, ATP, the primary energy currency of cells, and their reported anti-inflammatory and anti-tumor effects. While still in the early stages of research, the study of cordyceps provides a fascinating window into the intricate connections between fungi and animal behavior, a reminder that the natural world holds countless secrets, waiting to be unveiled. The rainforest, and the seemingly simple fungi growing within it, demonstrate a power far exceeding our initial perceptions. The humid air of the Amazon rainforest hung heavy, thick with the scent of decaying leaves and damp earth, a perfect environment for a multitude of secrets. Beneath the towering trees, a world of intricate beauty thrived, a world largely unseen, dominated by an astonishing group of fungi called Ascomycota. This wasn't a singular, simple kingdom of mushrooms, as many might first assume. It was a breathtaking tapestry of life, encompassing a far greater variety of forms and functions than any other fungal division. Ascomycota, derived from the Greek word askos, meaning o cup, are defined by a very special way they reproduce through asci. These asci are tiny, sac-like structures, and within each one, spores are formed, ready to be released and carried to new locations. Unlike the familiar Basidiomycota, which produce spores on specialized club-shaped structures, the Ascomycota rely almost exclusively on this cup method. This difference in reproduction significantly shapes their diversity, leading to an incredible array of lifestyles and adaptations. Consider the yeasts that quietly ferment sugars in our breads and beers. Saccharomyces cerevisiae is a classic example. These single-celled ascomycetes, though simple in appearance, are utterly vital to human civilization, representing a tiny sliver of the vastness within this group. But the story doesn't end there. The Ascomycota stretch across an unbelievable range of forms. There are the powdery mildews that bloom across rose bushes, causing a delicate white fuzz, and the chestnut blight that devastated American chestnut forests in the early 20th century. These aren't simply destructive forces, they are fundamental players in ecosystems, mediating decomposition and influencing nutrient cycling. Moving into terrestrial environments, the diversity becomes even more striking. Many ascomycetes are exceptionally important in plant ecology. Morels, those delectable mushrooms prized by foragers, belong to this group, relying on a symbiotic relationship with specific tree roots, typically oaks and beeches, to access nutrients. Similarly, tucastics, found in the alpine regions of New Zealand, form long, slender associations with the roots of native grasses, forming a crucial link in the plant's ability to thrive. These relationships demonstrate the intricate ways ascomycetes contribute to the health and resilience of plant communities. The Ascomycota also hold the key to some remarkably sophisticated strategies for dispersal. Geomyces glutinum, for example, produces a stunning, iridescent powder, a shimmering, silver dust that it strategically releases into the air. This isn't just a beautiful spectacle. The spores are incredibly light and aerodynamic, allowing them to be carried vast distances by even the slightest breeze, effectively colonizing new habitats. Other ascomycetes utilize specialized structures, like fruiting bodies that mimic the appearance of other plants to attract pollinators, or rely on explosive mechanisms to launch their spores with considerable force. The ecological roles of ascomycetes extend far beyond simple decomposition. Some species are masters of chemical warfare, producing toxins to inhibit competitors, while others secrete enzymes that break down incredibly tough materials like lignin, the complex polymer that makes up plant cell walls. This ability to degrade lignin is crucial to the decomposition process, allowing the nutrients locked within dead plant material to be returned to the soil, making them available to new generations of plants. Furthermore, the diversity within the Ascomycota includes some genuinely bizarre and fascinating organisms. A tuber melanosporum, the black truffle, is a prime example. Its subterranean mycelial network develops a remarkable sensitivity to the presence of tree roots, leading the fungus to grow directly beneath them, essentially farming the roots for their nutrients. Then there are the more enigmatic lichen, a symbiotic partnership between an ascomycete and a green algae or cyanobacteria. The ascomycete provides a stable structure and protection, while the algae or cyanobacteria performs photosynthesis, providing the fungus with sugars. The genetic diversity within Ascomycota is equally astonishing. It's estimated that they represent roughly 30% of all known fungal species, illustrating the sheer scale of their evolutionary success. Research continues to uncover new and previously unknown species, revealing even more complex adaptations and ecological roles. Their evolutionary history is intricately woven into the fabric of life on Earth, and their continued contributions to global biogeochemical cycles are profoundly important. As you drift off to sleep, consider the quiet power and astonishing variety contained within this seemingly small group of fungi, the Ascomycota. Their story is a testament to the boundless creativity of evolution, a story of collaboration, adaptation, and a fundamental role in the health and balance of our world. Their existence, often unseen and unnoticed, quietly sustains life as we know it. The scent of damp earth, rich and slightly sweet, drifted through the air as Professor Armitage adjusted his spectacles, a thoughtful frown creasing his brow. "You see, children," he began, his voice a low, comforting rumble. "For centuries, humanity has wrestled with the problem of pollution. We've built incredible machines, developed sophisticated chemical treatments, and poured vast resources into cleaning up contaminated land and water. But sometimes, the most elegant solutions are hidden in the quietest corners of the natural world, specifically within the kingdom of fungi." He gestured towards a large, intricately patterned display showcasing different fungal species, each thriving in a carefully controlled microcosm mimicking various polluted environments. The concept behind what we call mycoremediation is remarkably simple, though the science underpinning it is profoundly complex. It's about harnessing the incredible metabolic capabilities of fungi to break down and absorb toxins, effectively transforming a hazardous site into a healthier one. The key to this process lies in the extraordinary enzymes produced by fungi. These enzymes are like tiny, highly specialized molecular scissors, capable of dismantling complex organic molecules, pollutants, into simpler, less harmful substances. Unlike many chemical treatments, which often create new, unintended byproducts, fungal enzymes are naturally geared toward breaking down complex molecules like petroleum, pesticides, heavy metals, and even plastics. Professor Armitage pointed to a particularly robust-looking species, Iris pleurotus ostritus, commonly known as oyster mushroom, growing within a tank containing a simulated oil spill. This particular fungus, and many others, have evolved over millennia to thrive in environments rich in hydrocarbons. They don't just tolerate the presence of oil, they actively consume it. They secrete enzymes that effectively digest the oil, transforming it into water, carbon dioxide, and biomass essentially, turning a persistent pollutant into something harmless. The beauty of mycoremediation is that it's often a remarkably passive process. Once the fungi are introduced to a contaminated site, they begin to colonize the area, establishing a network of hyphae. These are like incredibly fine thread, like filaments that spread throughout the soil, water, or even the fabric of a polluted material. As the hyphae grow, they come into contact with the pollutants and begin the breakdown process. In some cases, scientists can accelerate this process by providing the fungi with optimal conditions, moisture, warmth, and a readily available food source. However, mycoremediation isn't a one-size-fits-all solution. The effectiveness of a particular fungal species depends heavily on the type of pollutant it's encountering and the specific environmental conditions. Some fungi are specialists, capable of breaking down only a narrow range of contaminants. For example, certain Phanerochaete chrysosporium strains known for their role in bioremediation of nuclear waste have demonstrated remarkable abilities in degrading radioactive isotopes. Others, like S. tremens versicolor, the artist's mushroom, are particularly adept at breaking down plastics, a challenge that is becoming increasingly urgent with the ever-growing accumulation of plastic waste globally. The process often involves a two-stage approach. Initially, the fungi break down the large, complex pollutants into smaller, more manageable molecules. Then, other microorganisms, bacteria, in particular, take over, further degrading these intermediate compounds into harmless substances like water, carbon dioxide, and nitrogen. It's a symbiotic relationship, a carefully orchestrated dance between different organisms, all working together to restore balance. Beyond simply breaking down toxins, fungi also demonstrate an ability to immobilize heavy metals. For instance, some Cladosporium species can bind heavy metals like lead and cadmium to their hyphae, preventing them from leaching back into the soil or water supply. This process effectively sequesters the pollutants, rendering them harmless and protecting surrounding ecosystems. Scientists are experimenting with a range of techniques to optimize mycoremediation. In some cases, they're inoculating contaminated soil with a carefully selected blend of fungal species, tailored to the specific pollutants present. In other cases, they're using mycelial mats, thick layers of fungal growth, to contain and treat contaminated areas. Innovative approaches even include using fungi to grow bricks or building materials from contaminated soil, effectively incorporating the pollutants into a stable, usable product. The potential of mycoremediation extends far beyond simply cleaning up traditional industrial pollution. It holds promise for addressing emerging environmental challenges, such as microplastic contamination and the accumulation of pharmaceuticals in waterways. As our understanding of fungal diversity and their metabolic capabilities continues to grow, so too will the sophistication and effectiveness of this remarkable process. Professor Armitage paused, a gentle smile gracing his face. The Earth, children, is full of solutions often hidden in plain sight. And sometimes, the most powerful tools are the smallest, most unassuming ones, the quiet, persistent, and incredibly adaptable fungi. He gestured again to the display, a silent invitation to ponder the extraordinary potential of this hidden kingdom. The forest floor wasn't silent, not really. It pulsed with a quiet, insistent activity, a vast, hidden network working tirelessly beneath the fallen leaves, decaying wood, and the remains of ancient trees. This intricate dance was orchestrated by a remarkable group of fungi known as saprophyphous fungi, the specialized decomposers. They weren't flashy like some of their relatives, building elaborate structures or producing stunning displays of color. Instead, they were masters of subtle transformation, relentlessly breaking down organic matter with remarkable efficiency. These fungi exist as a complex web of hyphae, microscopic threads that spread out across the soil and wood. Think of them as tiny, branching explorers, each one seeking out a nutrient-rich meal. Unlike many fungi that form fruiting bodies, the recognizable mushrooms we often see, saprophyphous fungi primarily live as this hyphal network, a vegetative state dedicated to the vital task of decomposition. The hyphae themselves possess powerful enzymes, essentially biological scissors, capable of dismantling complex molecules like cellulose, lignin, and chitin, the very building blocks of plants and insect exoskeletons. The process began with a gentle assault. The hyphae secreted enzymes into the surrounding material, slowly dissolving the intricate structures. Cellulose, for example, which makes up the cell walls of plants, is remarkably resistant to breakdown. But the enzymes produced by saprophyphous fungi, particularly cellulase, were specifically designed to cut through it. Lignin, the tough, woody component of trees, presented an even greater challenge, requiring a combination of enzymes and sometimes the assistance of symbiotic bacteria. The rate at which decomposition occurred varied greatly depending on the type of material and the specific saprophyphous fungi involved. Some species were particularly adept at breaking down soft wood, while others specialized in decaying leaves or even the bones of long, dead animals. Scientists have identified hundreds of different saprohyphus fungal species, each with a unique set of enzymatic capabilities and, consequently, a specialized role within its ecosystem. The impact of these fungi on ecosystems is profound and fundamental. Without them, the world would be buried under an unyielding blanket of organic debris. Imagine a forest where fallen leaves remained intact for centuries, preventing new seedlings from sprouting. Or a landscape where ancient trees never returned to the earth, their carbon locked away indefinitely. Saprohyphus fungi are the crucial link in a continuous cycle, a cycle of death and rebirth. They don't simply eat dead material. They recycle it. The products of their breakdown sugars, amino acids, and mineral nutrients are released back into the soil, providing nourishment for plants and other organisms. This is a kind of fungal fertilization, enriching the soil and making it available for new growth. It's a sophisticated exchange, a silent partnership between the fungi and the plants, where both benefit from the transformation. The activity of saprohyphus fungi also plays a key role in carbon cycling. As they break down organic matter, they release carbon dioxide into the atmosphere. However, this release is carefully regulated, and the fungi play a role in sequestering carbon within their hypha, temporarily storing it until it's released later. This intricate balance is a vital component of the global carbon cycle, influencing the climate in subtle but significant ways. Interestingly, the diversity of saprohyphus fungi within a particular forest ecosystem is closely linked to the diversity of the decaying material available. A forest dominated by beech trees, for example, would harbor a different suite of saprohyphus fungi compared to one with a mix of oak, pine, and maple trees. Each fungal species has evolved to thrive in a specific niche, contributing to the overall resilience and health of the forest. Furthermore, the impact of saprohyphus fungi extends beyond simply recycling nutrients. They are involved in complex interactions with other organisms. Some fungi form symbiotic relationships with tree roots, exchanging nutrients and water for carbohydrates, a relationship known as mycorrhizae. While mycorrhizal fungi are often considered a separate group, many saprohyphus fungi also engage in similar exchanges, demonstrating the breadth of their ecological influence. The study of saprohyphus fungi is revealing ever more astonishing details about the hidden workings of the natural world. Scientists are discovering that these fungi are capable of communicating with each other through networks of hyphae, sharing information about nutrient availability and potential threats. This wood wide web, as it's sometimes called, suggests a level of interconnectedness within ecosystems that was previously unimaginable. As the sun begins to set, casting long shadows across the forest floor, the activity of the saprohyphus fungi continues, diligently transforming the landscape, one tiny hypha at a time. Their work is a constant reminder of the fundamental importance of decay, of the necessity of transformation, and of the remarkable resilience and interconnectedness of life itself. It's a quiet, unassuming story, but a story that is essential to the health and balance of our planet. The story of fungi is, at its heart, a story of incredible adaptation, a tale of survival honed over hundreds of millions of years. For a long time, we've understood fungal reproduction as a relatively simple process, spores simply floating away, and hopefully landing somewhere good. But the truth is far more intricate, a process steeped in genetics and driven by an ancient pressure, ensuring the best possible offspring. The emergence of sexual reproduction in fungi wasn't a sudden event, but a gradual unfolding, a series of refinements that ultimately provided a dramatic advantage. It began with a fundamental shift, the introduction of meiosis. Before meiosis, fungal reproduction was largely asexual, relying on mitosis, a process that creates identical copies of the parent. While effective for rapid colonization and spread, Mitosis lacked the critical element of genetic diversity. Imagine a field of fungi, all producing spores with the same traits. If the environment changed, perhaps a new disease appeared, or a nutrient source dwindled, all these clones would be equally vulnerable. Meiosis, however, offered a solution to this inherent instability. It's a type of cell division that halves the number of chromosomes, shuffling the genetic deck. The precise origins of meiosis in fungi are still being actively investigated, but the prevailing theory suggests it arose in a group known as the Zygomycota, a diverse collection of fungi that includes bread molds and some plant pathogens. These fungi, early in their evolutionary journey, began to express genes that allowed them to undergo meiosis, a complex process involving paired chromosomes and a reduction in genetic material. This wasn't a simple switch. It was a gradual accumulation of genes that ultimately enabled this remarkable division. The process begins with a eukaryotic cell, a cell containing two complete sets of chromosomes, one from each parent. This stage is crucial. It allows for a controlled mixing of genetic information, a sort of internal remixing of the parental genomes. The formation of the eukaryotic cell is intricately linked to a specialized mating type. Most fungal groups, including the Zygomycota, possess distinct mating types, often designated A and B. These are not simply morphological differences. They represent genuine genetic distinctions, dictated by the expression of specific genes. When two fungi of different mating types come into contact, a process called plasmogamy occurs. This is where the cytoplasm, containing the cellular machinery of the two fungi, merges, creating a eukaryotic cell. It's a meeting of equals, each contributing its genetic resources to the nascent combination. Following plasmogamy, the eukaryotic cell then enters a phase called karyogamy, where the two nuclei, each containing a full set of chromosomes, finally fuse together. This creates a diploid cell with two sets of chromosomes. However, this diploid state is typically short-lived. The diploid nucleus rapidly undergoes meiosis, restoring the haploid state and producing 4 haploid spores. These spores, unlike those produced through asexual reproduction, carry a unique combination of genetic material from both parental fungi. The spores themselves are marvelously engineered structures. Their walls are incredibly tough, shielding the precious genetic material within from damage and allowing them to withstand harsh conditions. They're not just passive carriers of genes, they're miniature survival capsules. The spores released from a fungal fruiting body, the visible reproductive structure, are then carried by wind, water, or animals, dispersing the genetic potential across the landscape. The spores don't just float aimlessly. Their formation is influenced by a sophisticated signaling system, often involving chemical compounds. These compounds act as mating signals, guiding spores towards individuals of the opposite mating type. This directed dispersal is remarkably efficient, increasing the chances of successful fusion and the creation of new, genetically diverse offspring. The evolution of sexual reproduction wasn't just about producing new spores. It was about bolstering the fungi's resilience. Genetic diversity within a population provides the raw material for natural selection. If a disease appears, some spores carrying genes that confer resistance will be more likely to germinate and produce a thriving fungus. Similarly, if a change in the environment favors a particular trait, the spores carrying those genes will have a significant advantage. Over countless generations, this interplay between genetic diversity and environmental pressures has shaped the incredible diversity of fungi we see today. The complex system of mating types, plasmogamy, karyogamy, meiosis, and spore dispersal represents a finely tuned mechanism for maximizing the chances of survival and adaptation. It's a testament to the power of evolution, a story written in the very fabric of fungal life. a process that continues to unfold with each new generation of spores carried on the wind. The humid air of the rainforest floor, thick with the scent of decaying leaves and damp earth, wasn't just a place of immense beauty. It was a giant, ongoing laboratory. For millennia, fungi have been quietly rewriting their own genetic code, responding to the ever-shifting conditions around them. We've only recently begun to understand the astonishing complexity of this process, what scientists are calling fungal genetics. And it's revealing just how remarkably adaptable these organisms are. It's not the straightforward inheritance we see in plants or animals. Instead, fungal genetics operates on a far more flexible, dynamic level, driven by the need to survive and thrive in a constantly changing world. The key difference lies in the way fungi store and utilize their genetic information. While animals and plants have DNA neatly packaged into chromosomes within a nucleus, fungi largely lack a true nucleus. Their genetic material, known as the nucleomorph, is dispersed throughout the cytoplasm, a gelatinous substance that fills the fungal cell. This doesn't mean their genes are less important. It simply means they're more readily accessible and more easily modified. This fluidity allows for a process called horizontal gene transfer, a remarkable ability where fungi can exchange genetic material directly with other fungi, even those from entirely different species. Scientists have discovered intricate mechanisms that facilitate this exchange. One method involves bacterial shuffling, where fungi effectively borrow genes from bacteria that they encounter. These genes often relate to metabolic pathways, the intricate networks of chemical reactions that allow fungi to break down complex compounds. For example, a fungus might acquire a gene from a bacterium that allows it to digest a new type of wood, quickly gaining a competitive advantage in its environment. Another method, called cell fusion, involves two fungal cells merging their cytoplasm, essentially creating a hybrid with a combined genetic makeup. This isn't a simple mix. The two genomes can rearrange themselves, creating entirely new combinations of genes. Researchers have meticulously tracked these exchanges, using advanced genomic sequencing techniques to identify the genes being transferred and the pathways they influence. They found that fungi regularly swap genes related to stress response, allowing them to rapidly adapt to harsh conditions like drought, heat, or nutrient deprivation. A fungus facing starvation, for example, might acquire genes that enable it to utilize a completely new food source, such as a previously inaccessible polysaccharide. This isn't a conscious decision. It's a purely genetic response, driven by the organism's innate need to survive. The study of fungal genetics has also illuminated the role of transposons, mobile genetic elements that can jump around within the fungal genome. These transposons act like little repair crews, inserting themselves into new locations in the genome and potentially altering gene expression. They can activate dormant genes or silence existing ones, providing a powerful mechanism for rapid adaptation. Scientists have observed transposons moving hundreds or even thousands of base pairs, the building blocks of DNA within a single fungal generation. This level of genetic shuffling is astounding, and it's driving much of our understanding of fungal plasticity. Furthermore, the research isn't just about understanding how fungi adapt to their immediate surroundings. Scientists are beginning to uncover genetic pathways that allow fungi to respond to long-term environmental changes, such as climate shifts. Some fungi, for instance, have evolved genes that enable them to tolerate higher temperatures, reflecting the increasing heat stress experienced by ecosystems worldwide. Others have developed genes that allow them to break down recalcitrant compounds, substances that are difficult to decompose, hinting at the potential for fungi to play a role in mitigating the effects of plastic pollution. The tools used to study fungal genetics are becoming increasingly sophisticated. Scientists are utilizing Cas9 technology, a precise gene editing tool that allows them to target and modify specific genes within a fungal genome. While initially developed for plant and animal research, CRISPR is now being applied to fungi, allowing researchers to directly investigate the function of specific genes and to create fungal strains with tailored adaptations. These engineered fungi could potentially be used for a wide range of applications, from bioremediation to sustainable materials production. The ongoing research into fungal genetics isn't simply about cataloging adaptations. It's unveiling the fundamental principles of evolutionary change. It demonstrates that fungi aren't just passive responders to their environment. They're actively shaping their own evolution, utilizing a remarkable genetic toolkit to navigate the challenges of a changing world. As we delve deeper into this fascinating field, we're beginning to appreciate the immense potential of these tiny organisms and the profound impact they have on the health and stability of our planet. The complexity and adaptability of fungal genetics is a testament to the power of life's ability to find a way, even in the most unexpected of places. The forest floor, perpetually damp and cool beneath the ancient canopy, wasn't just a landscape of decaying leaves and wood. It was a riot of hidden color, a spectrum far richer than the brilliant greens and browns we typically associate with the wild. This hidden vibrancy came from the fungi, and not just from the familiar earthy browns of many mushrooms. Scientists are increasingly uncovering an astonishing array of pigments within the fungal kingdom, a palette that's reshaping our understanding of their roles in ecosystems and even hinting at their evolutionary history. For a long time, the color of fungi was largely overlooked, a consequence of simply observing the mature fruiting bodies. Researchers focused primarily on the brown hues produced by melanin, a pigment also found in human skin and many other organisms. Melanin is a fairly robust pigment, responsible for protecting fungi from UV radiation and perhaps contributing to the structural integrity of the mushroom. However, as technology has advanced, particularly in the realm of microscopy and molecular analysis, we've begun to realize that fungi hold a far more diverse collection of color compounds. One particularly fascinating group of pigments are the melanins, but they're not all the same. There are variations in their chemical structure, each producing a slightly different shade of brown, from deep chocolate to pale cinnamon. Some melanins, notably those found in mycelial species, produce an intense, almost iridescent dark brown, a testament to their intricate molecular architecture. But beyond these browns lie yellows, reds, oranges, even blues and purples, a breathtaking display revealed only through careful observation and specialized techniques. The production of these diverse colors isn't simply a cosmetic trait. It's deeply linked to the fungi's function. For instance, the bright orange pigments found in Amanita muscaria, the fly agaric, aren't just a result of genetic variation. These pigments, primarily carotenoids, compounds also found in carrots and many fruits, provide significant protection against UV damage. In a similar vein, the vibrant red pigments in certain Russula species demonstrate a critical role in deterring herbivores. The bright color serves as a potent warning signal, indicating that the mushroom contains toxic compounds, a chemical defense mechanism honed over millennia. Beyond defense, pigments are also involved in communication. Some fungi use pigments to attract insects, specifically the flies that are crucial for spore dispersal. The orange of Amanita muscaria, for example, isn't just a deterrent to animals that might eat it, but a lure for flies, who are then unwittingly carried to new locations as the mushroom releases its spores. The red pigments in other species likely play a similar role, attracting specific insects to facilitate reproduction. But the discovery of pigments like phycobilins, often associated with algae and cyanobacteria within certain fungal species, is truly remarkable. Phycobilins are responsible for the brilliant blues and purples seen in some mycena varieties, and their presence suggests a possible symbiotic relationship with photosynthetic microorganisms. These fungi may have evolved to host algae within their tissues, receiving nutrients from the algae while, in turn, providing a protected environment for the microorganisms. This represents a fascinating example of co-evolution, where two organisms have become inextricably linked through the shared use of a pigment. Furthermore, researchers are investigating pigments called flavonoids. These compounds are known for their antioxidant properties, and their presence in fungi suggests they contribute to protecting the organism from oxidative stress damage caused by free radicals. The production of these pigments likely reflects the fungi's ability to thrive in challenging environments, where exposure to damaging elements is frequent. The study of fungal pigments isn't just about identifying new colors, it's about reconstructing fungal evolution. The distribution of these pigments across different fungal groups can provide clues about their ancestry and how they adapted to diverse ecological niches. Certain pigments are remarkably conserved across fungal lineages, suggesting they were present in the earliest fungal ancestors. Conversely, the presence of unique pigments in specific groups indicates that those groups have undergone significant evolutionary diversification. New techniques, such as hyperspectral imaging, allow scientists to capture a far more detailed spectrum of light reflected by fungi, revealing subtle color variations that would be invisible to the naked eye. This technology is opening up entirely new avenues of research, allowing scientists to analyze the color patterns of fungi across vast landscapes. The ongoing exploration of fungal pigments is unveiling a complexity and beauty within the fungal kingdom that was previously hidden. It's a reminder that even the most familiar landscapes hold secrets, waiting to be discovered through careful observation and scientific inquiry. These colors aren't merely aesthetic, they are functional, communicative, and integral to the very fabric of fungal life, a vibrant testament to the remarkable adaptability and diversity of the fungal world. The scent of damp earth and decaying leaves, a familiar aroma across countless landscapes, holds more than just the ghosts of seasons past. It's a smell increasingly recognized as a key to a remarkably hopeful future, a future interwoven with the astonishing potential of fungi. For centuries, fungi were largely dismissed as simple decomposers, the shadowy servants of the natural world. But recent scientific breakthroughs are revealing a world of astonishing complexity and capability, poised to revolutionize medicine, manufacturing, and even how we think about sustainability. The most immediate and perhaps profound changes are unfolding in the realm of medicine. We've long known about penicillin, a discovery that dramatically shifted the course of human health. But that was merely a single star in a burgeoning constellation of fungal therapies. Scientists are now isolating and studying compounds from a bewildering array of fungi with powerful antiviral, antibacterial, and even anticancer properties. Ganoderma lucidum, or reishi mushroom, isn't just a traditional tonic. Researchers are examining its polysaccharides, complex sugar molecules, for their ability to stimulate the immune system and potentially slow the progression of cancers. Tremites versicolor, commonly known as turkey tail, is gaining traction as a complementary therapy, demonstrating promising results in boosting immune responses during and after chemotherapy. The beauty of this approach isn't just in the potent compounds themselves, but in the relatively simple, sustainable methods of cultivating these fungi in controlled environments. Unlike traditional pharmaceutical manufacturing, which often relies on energy, intensive processes, and complex chemical synthesis, Fungal cultivation requires comparatively little energy and can be scaled up rapidly. Beyond established therapies, a new wave of research focuses on fungal enzymes. These tiny biological catalysts are remarkably efficient and can perform reactions that are difficult or impossible to achieve with conventional chemical processes. For example, certain fungi produce enzymes capable of breaking down plastics, a monumental challenge for the planet. Scientists are exploring ways to use these enzymes, combined with microbial consortia, to effectively eat plastic waste, potentially offering a true solution to the accumulating global plastic crisis. The key here is not just the ability to degrade the plastic, but the ability to do so at ambient temperatures and pressures, drastically reducing the energy demands of recycling. However, perhaps the most transformative applications of fungi lie in the field of materials science. The idea of building with mushrooms, a concept once relegated to novelty shops, is rapidly becoming a serious industry. Mycelium, the root-like structure of fungi, is incredibly strong, lightweight, and remarkably versatile. It's composed of chitin, a complex carbohydrate also found in insect exoskeletons, which provides exceptional tensile strength. Researchers are now using mycelium to create everything from building materials to furniture to packaging. One particularly exciting area is myco-architecture. Companies are growing mycelium within prefabricated molds, shaping it into bricks, panels, and even complex architectural forms. What's incredible is that the mycelium binds the material together, eliminating the need for traditional adhesives and significantly reducing the carbon footprint associated with construction. These mycelium-based materials are not only sustainable, but they are also biodegradable, returning to the earth naturally at the end of their lifespan. Furthermore, mycelium is being explored for its potential in creating leather alternatives. By cultivating mycelium on agricultural waste, scientists can generate a material with a texture and appearance remarkably similar to genuine leather, without the ethical and environmental concerns associated with animal agriculture. The ingenuity doesn't stop there. Researchers are experimenting with mycelium as a binder in composite materials, dramatically enhancing the strength and durability of products like carbon fiber. This is particularly relevant in the aerospace industry, where lightweight, high-performance materials are crucial. Another innovative use is in creating packaging materials, mycelium-based cushioning that protects fragile goods during shipping, again significantly reducing the reliance on petroleum-based plastics. Looking even further ahead, the complexity of mycelium is inspiring new approaches to materials design. Scientists are learning to program mycelium growth, guiding its branching and density to create materials with specific properties. Imagine creating a material that becomes stronger as it ages, or one that responds to changes in temperature or light. These aren't science fiction concepts. They're increasingly within reach, thanks to our growing understanding of fungal biology. The future of fungi isn't just about replacing existing materials. It's about fundamentally rethinking our approach to design and manufacturing. It's about creating a closed-loop system where waste is minimized, resources are used efficiently, and materials return to the earth naturally. The quiet, unassuming power of fungi, once overlooked, is poised to reshape our world, a testament to the profound interconnectedness of life on Earth and the remarkable potential hidden within the most unexpected corners of our planet. It's a future built not on extraction and dominance, but on collaboration and regeneration, a future profoundly shaped by the wisdom of the mycelial network. Let the memories of the forest, of silent growth and hidden wonders, settle within you. As we conclude our journey through the world of fungi, allow your mind to quiet, to find gentle stillness. Let your thoughts drift, calm and unhurried, until sleep embraces you.